Mike and Me podcast. Just want to bring you, I don't know, just for me, I talk about AI a lot, but here's just unedited. You know, you can find this on YouTube or Google Deepmind, their channel. But yeah, this is valuable, fascinating information that you can I see promising, and also you can read the faces. It's just fascinating to look across the faces, especially the host. You can read his face and his body language and how you want. And yeah, either way, this is very important for those that are even curious, because most AI news and information you're gonna see is economically what makes sense, pointed, and but it's just it's really cool to see some of the actual scientists' point of view that are actually implications for health and just humanity in that regard. So, yeah, here you go.
This panel has to do with the frontiers of science. And I've interpreted, and we've interpreted, we had a conversation before, interpreted the word frontiers very broadly. Obviously, one of the frontiers of AI is the human. Um, Another frontier of AI is what is going to be done with all the knowledge that is being collected and how that knowledge influences discovery. So there's a question of discovery being the frontier, and the management of all of these incredibly powerful tools is also a frontier. Um, um, which leads us to the question of what is a human. A human is, to some people, a human is a locus of information. A human has personhood. We experience grief, joy, sorrow. We are patients. We are caretakers of patients. We suffer when we have diseases. We suffer when our friends and family and loved ones have diseases. We have privacy. We have dignity. We have empathy. And we are vulnerable. So there are many other things, many other vectors. We also discover new things. And when we discover new things, we are challenged because we have to figure out what to do with those discoveries. So the way I thought that we would run this panel is to first talk for each one of us to talk about how we use AI in discovery, in our particular discovery, to be very specific about how we use AI in our discovery, and a little perspective on how our each of our fields is working with the discovery and AI. The second aspect I want to talk about is to veer a little bit into controversy, and ask the question, because I think that it is almost inevitable that, at least in my lifetime, that there will be some version of an AI Fukushima. And by that, I mean there will be a breach of data, or a breach of privacy, or a discovery that takes us to the brink of something that's unpalatable, that's dangerous. There may be there's already been, in the recent elections, the use of AI, not in our sphere, but in the use of AI as a weaponized mechanism of false information. So that brings us to another frontier, which is the frontier of how to take all of this and potentially mitigate against these problems. So that's going to be the broad overview of what we're doing. And then finally, we'll talk more optimistically about the future in each of our fields, and talk about what we think about as these fields move ahead, and how we can incorporate and while mitigating the risks. So I'll start with, I'll call everyone by their first name, if that's okay, because there are too many honorifics that follow. I'll start with Anna, and maybe we can start with you talking about what you do and how you incorporate AI in your work and what the field is looking at.
Okay, thank you. Yes, my work is to be a pathologist, but I'm also the head of a new institute about women's cancer. So first, starting with pathology, AI, we have been validating or co-developing tools with different startups to have tools enhanced to facilitate the diagnosis. And also to challenge a question, can we predict a BRCA1 and 2 status from H&E slides? This was done with an academic lab, head being Thomas Walter from the École des Mines, PSL University. And so finally, I found it very exciting because it opens for using tools for diagnosis. It brings us speed, but it has to be very accurate and efficient. The tool has to be accurate and efficient. But at the end of the day, I think that the human guarantee, because it will be, so far as I can forecast, always a pathologist who will sign out, it is a breast cancer. But in the Institute of Women's Cancer, which is an institute built by Institut Curie, PSL University, and INSERM, we really want to make new algorithms alone or with industry. And we hope to bring new tools that will facilitate our lives. But maybe we'll speak after how we are going to make that happen and accept it by the patients.
Sure, we'll come back to that.
Yeah. So I'm Janet Thornton. I'm a computational biologist, and I've worked in the world of protein structures. And I don't know if you saw at lunch that wonderful display of a protein structure rotating, that to me, they are some of the most beautiful things in the world. And so I started when we only had 20 protein structures. With AlphaFold now, we can model 214 million protein structures. So the change in the field, so if we're asking what impact will AI have on science, in this case, it had the most enormous impact. So that now structural biologists worldwide use this data to actually help them solve their structures and to validate their structures. And it's really transformed the field. Recent progress beyond the original AlphaFold has meant we can look at multimers and look at ligand binding. And of course, we know that proteins, which we're all made of, actually only work because they interact with other things. And so being able to look at and predict these interactions and the modulations that happen during evolution, to me, remains one of the most exciting and demanding areas of molecular biology. But going on to the cellular biology and understanding the human body as a whole and medicine in particular, then we will need to go to this next step of integrating the data for an individual cell and working out how that cell interacts with other cells. And that, I think, will be an enormous thing going forward, actually. Sure. It's the basis of our future understanding of life.
Great. Anna, tell us about what you do. And
I think that's a wonderful segue into my story. So I'm a physician scientist and a cell biologist, and of course, the 37 trillion cells or so in our bodies are really, it's the unit of life. And so understanding cells, I mean, I actually study membrane proteins. So proteins fascinate me by how they come together to organize themselves in cells and all the basic functions that basically allow us all to work, our systems and organs to work. So I'd like to illustrate how we use AI by telling you a story of scientific discovery that comes from my group. So I work at the Broad Institute, so it's a kind of a place where a lot of genetics and genomics happen on a daily basis. And so as a physician scientist, I've taken an interest on trying to sort of understand how misspellings in the DNA lead to broken circuits inside cells, and then, of course, how we find ways to fix them. So this particular story begins on the Mediterranean island of Cyprus, where it was actually known for many decades that there were many families in which in every generation several members suffered and died from what turned out to be a mysterious kidney disease. So the mystery was solved by the Broad Institute in that we actually found that this was because of a misspelling that was hiding in a very dark corner of the human genome in a variable number of tandem repeats region of a gene called mucin-1, which is a glycoprotein, a membrane protein. And then my team took on this challenge of figuring out how this misspelling results in broken circuits inside these kidney cells. And in order to do that, and that's the first example of how machine learning was used, and this is now for image recognition, essentially. I have calculated, because I had to sort of tally up all the work that we did, we used a lot of imaging-based approaches, as cell biologists do, but we did it at high throughput. And I think over the course of several years, we imaged what I have counted to be about 15 million cells that we basically were able to then assess how these circuits inside the cells are broken on account of this particular mutation. And then, of course, we also discovered a small molecule by doing this approach, again, image-based screening, in order to hopefully get to a therapeutic. And so what we found in doing this, and this takes us to the next use of AI, is that the misspelling in the DNA, the mutation, results in a misshapen protein that accumulates inside the cells. And this accumulation is caused by another molecule, what I call a nodal molecule. It turns out to be something called a cargo receptor. The details don't matter, but just to say that this is a system that captures misshapen, misfolded proteins and holds onto them so tight that the cells eventually die because there's too much accumulation of toxic waste, if you will. And then it turns out that this discovery is actually what I call a nodal mechanism, because it's not only relevant to this particular misfolded protein that causes kidney disease, but it turns out, as we discovered, it's also relevant to a misfolded protein that causes a form of Alzheimer's, and a misfolded protein that causes a form of blindness in the eye, and on in the liver and the lung. So it turns out to be a nodal mechanism that we can now target therapeutically for the benefit of many more patients than we previously thought possible. And how did we define the perfect fitting drug, if you will, for this particular nodal target? Well, AlphaFold, and this is a structure that I shared with Demis when we met last summer, was actually brilliant in figuring out how a heterotetrameric structure, so four things coming together, are actually forming a structure that creates a specific pocket for a drug that we discovered that is now disrupting this nodal mechanism and can therefore have therapeutic benefit for all the diseases that I mentioned. And I'm pleased to say that by using machine learning for images, and then ultimately AlphaFold for this target. We both did target discovery and then drug discovery that is now actually making its way toward patients in partnership with a pharmaceutical partner from my lab. And so this is just an example to illustrate the power of these tools that can only get better and better as the years go on. And so I'm very excited for what the potential for the future
So
will be.
pathology, genomics, basic discovery. I lie at the far end. I'm a drug discoverer and a clinical scientist. I have a quick fun story as well. My lab has been involved in making antibody drug toxins. These are antibodies that are conjugated to toxins. One of the things that we use, of course, is AlphaFold to figure out the structure of the antibody and where to potentially attach the toxins. And a very common drug in women with Herceptin-resistant breast cancer is a drug called Enhertu. It's been out for a while now, very commonly used. The problem with Enhertu is that it causes a 10 to 20% risk of interstitial lung disease, which we didn't know why, and 2 to 3% of those patients with ILD will die. And this is how sad and of course how desperate the field is, that people will take a 2 to 3% risk of death in order to get the benefit of Enhertu. So we were making, using AI tools, we were making these various drug toxin combinations and putting them into models to show ILD. And we asked the AI module basically to make all combinations So to create every possible combination. And one combination it threw out, which we said was a stupid idea, it threw out the combination of just the antibody with just the linker. And it said, you haven't really tested that part, have you? And we said, that's a really stupid idea. Of course, there's nothing. It's a linker. Linker is a passive element. It's the piece that links the chemical, to the toxin, to the antibody. Well, as case may be, turns out that in fact the linker is the toxic part. We're just publishing that data, and we would have not found that. So here's an example in which the AI module suggested an experiment that humans had to ingest and basically be humbled by, and then find out that in fact that was the right experiment to do. It was a control that a smart graduate student would have suggested. We just neglected to have done it. So across the spectrum, again, an incredible use of AI. Let's talk about data, because a lot of the conversation has been focused on data. And when I think about data, I think about three corners. And we talked a little bit about this earlier. One corner is what I call access quality integration. So I'd like all of us to take each of these corners and reflect a little bit about this in your own fields. The second is privacy. And the third is equity. So again, access quality integration, equity, and privacy. In pathology, in discovery, in genomics, how do you think about these corners? You don't have to tackle all three of them. Pick one, pick two. But how do you think about these? Because these are the frontiers that we will be facing or are already facing as our science interfaces with AI. Anna.
Yeah. For pathology, the situation is really different from within different countries. Here in England, the government decided to make digitalization accessible to a lot of pathology department. And in some other countries, for example, like in France, it started but very slowly. So for people to understand, glass slides from tissues that are biopsies or sample from surgical specimens were analyzed under a microscope to make diagnosis, using proteins to address the different diagnoses and the different therapeutic targets. But now we digitalize the slides, but it costs a lot to make a transition from a normal pathology lab to a fully digitalized lab. So here there is a bottleneck, because to make that happen, either it's a political decision and the government helps pathology departments if it's appropriate with regard to the economy and political organization of the country, or it's institution by institution that the decision is made. So this is the difficulty. And then to make big data sets for foundation models, we need a lot, 1 million of digitalized slides. So it means a lot of human work to go and retrieve the glass slides from trays where they are stored. It's an enormous work. So it's better to do it prospectively, on a daily basis, but to do retrospective analysis and to have huge data sets, it's really a tough job to do. Yeah.
But let me ask you a question.
Yeah.
Who owns that data?
Well, I think that's where the interpretation of the law may be different also country by country. If I'm asked the question, and this is the case, I would like to say, well, the glass slide comes from the body of the patient. So why isn't it the patient that owns his own tissue blocks, glass slides? But, for example, in France, it's part of the patient file. So it belongs to the hospital data warehouse. So I think that here, maybe lawyers and people who write the laws could have conversations with patients, advocates, patients, and pathologists, maybe. It could be helpful, I think.
Yeah, I mean, it seems to me that one of the things that we've glossed over in the data conversation is ownership. And I think it's very important to not gloss over that, because that's where the public distrust begins. And I'll give you a particular example. I want to talk about it in genomics as well, because it's very relevant. But as you know, the New York Times is currently suing OpenAI for the use of all its data. Writers are suing OpenAI for the use of, or other aggregate AI modules for the use of their data. Whereas, and so I gave you, we were talking a little bit beforehand, I said, well, on one hand, it's when you give a patient a consent form and you say, this, your data, donate your data because it might help other breast cancer patients in the future, most patients will say, absolutely, we'll do that. On the other hand, if that same consent form says, donate your data so that it can go to a company that begins with G, and they will then potentially monetize that data, that patient may not be so happy. So the question of who owns the data suddenly becomes much more complicated than just giving up data for the greater public good, because it is in fact usually aggregators of data that are getting a lot of the good. Janet, what do you think about data and privacy?
Data and, well, let me say something about data first.
Yes.
So I was director of the European Bioinformatics Institute that keeps curates, collects, etc., the data on molecular biology and beyond. So we had a lot of discussions around this. Personally, I was involved in the Protein Data Bank, the Worldwide Protein Data Bank, which has been through many years of discussing, actually, privacy at the beginning, because in the early days, it often took 5 years to determine a protein structure. And so crystallographers were very reluctant to share their data.
Yes.
They were very reluctant. And frankly, it took 20 years for everybody to come round to the idea that by sharing the data...
So give us a history lesson, Janet. How did they come around? How did the crystal... I mean, otherwise you wouldn't have AlphaFold, right?
No,
PDB.
we wouldn't. I should have made that point. Without the data, we won't have any AI. Or
Correct.
Any powerful in AI.
What was
so, in fact, the International Union of Crystallographers passed a statement saying that unless people deposited their data, they would not be able to publish in various journals.
It was not carrot, it was stick.
This was well, but there was also, yes, it was basically a stick. But many of the community were already on board. But there were some outstanding exceptions. Some of the best people actually didn't want to share their data. And so there needs to be a change in culture for the medical data, within the clinical profession actually, that they see the value, and in terms of privacy, that we can reassure people that the tools we have for anonymization or pseudo-anonymization are really powerful, because that is often the most challenging thing. I think also the Worldwide Protein Data Bank is publicly available for everyone. The data are essentially free. And I think this brings, personally, even for medical data, I would suggest that this, because if you have the knowledge of a given mutation and how to treat it, that's knowledge. You publish it, it's in...
Absolutely.
And you want to share that data as far as you can. So I would make a strong plea for public data, even if it's taken you 5 years or more to determine it, that there comes a time when where you can, and I think should, because most of the academic data, it's different if it's in a company, you can decide we will collect this data and this is our data. But actually, if it's publicly funded, I think most people would agree. And I think here in the UK, we have, because we have our National Health Service, there's a terrific opportunity if we can harness that data. And there's something called health data research, which is trying to promote and facilitate the sharing of data with the NHS, and we have our secure data environments in each hospital now. So this is a long process. As I said, in the crystallographic world, which is a tiny little world actually, it took 20 years to do this. For the health world, which is infinitely bigger and infinitely more complex, it's just going to take a long time to set up this ecosystem of data resources and access that allows people to access it. But this is all with the consent of the patient where possible, with understanding and education for the patients about how this will work and that they will, and for the doctors actually. There is a recent report on health data from Kathy Sudlow that was published last Friday, launched last Friday, and it does a beautiful description of the landscape of data in the UK. And it's really important that the government and all the many hospitals and everything really get together, because I do believe for infrastructure, it's kind of top down.
Right.
Research
Anna, I had a particular question, but take.
Well, I just wanted to comment that, we may not have to reinvent the wheel. After all, we have, as a community of scientists, sequenced by this time millions of humans, right? And this is all de-identified data that is federated. We have developed systems that can actually do that. We have done it with human genomes. James mentioned the pangenome this morning. There's, other aggregators where these data are de-identified, but then in a format that can be used by scientists all around the world. Now, that's just genomic information. What we're talking about here is multiple layers of information that already exist, however. I mean, if you took all the pathology labs on the planet, and aggregated all that data, we would probably have a lot of available data to train models for imaging, recognition, things like that. So I think that there's an opportunity to think about ways in which we can anonymize the data, but keep the metadata available and then federate the data in systems that could be shared, hopefully across governments and institutions. I know it's it's easier said than done, essentially, but we don't necessarily need to invent something that doesn't exist. We just need to build on what we've already done in order to, at the end, the bottom line is to help more patients. And so I'm hopeful that that can happen. But I didn't want to derail whatever.
No, I was continuing the conversation. I guess as we sort of begin to wrap up, I did want to talk about the fact that the word frontiers also means dangers. Every frontier also means encountering something new and unexplored, and therefore some of the concerns. And there are many we talked about. What are the mechanisms that you think about in your own fields about mitigating but potential risks, and take risk as broadly as possible, mitigating the potential risks that might lie ahead? What are the risks? What should we be watching out for before it happens? And how do we mitigate that? Maybe we'll start with Anna first and then go backwards.
Well, sure. I mean, we were just talking about, breach in privacy, patient privacy. Of course, that would be a concern, and that's the kinds of systems that we have to put in place for that to, to safeguard against that. I'm thinking about, AI can be used to develop bioweapons, for example. And that's something that, we have to face, and it's a real risk. I think that we need to use these tools responsibly. Again, I think meetings like this are a wonderful way to have this kind of dialogue about what is needed, and then what can be done to safeguard and mitigate the risks that exist. Any worthwhile human endeavor entails some risk, right? I think we can all agree on that. And so I think, shying away from it, I don't think that's a choice, but finding ways to use it responsibly. I was part of a conversation at the United Nations earlier this year about the same topic, among other topics, on the use of AI. And I think that there's a lot of other organizations and bodies that are now sort of trying to take this on. So that's encouraging. It would be nice to not over-regulate, but regulate just enough, which is always tricky, so that we do not stifle the scientific discovery, but also have some regulations in place to mitigate for such risks. Again, easier said than done, but I think this is where we are, and that's as practitioners of, using this tool in any of our scientific fields that we all represent here, I think it will be important to think about the balance between these two things.
Janet, when you began to see all these crystal structures being deposited, again, now turning towards discovery more than anything else, was there a concern that someone would use these crystal structures to build a new molecule that could be a toxin
well, yes, there was, but I think, as Anna said, and I agree with everything you said about you put in all the technical limitations, and protein design is obviously one of the things that
you can do, yes.
AlphaFold will enable, and that potentially can be dangerous. But I think 2 points I'd make. Obviously, you do all the technical things you can to avoid such issues. But perhaps more importantly, you educate the public, as Paul Nurse said. If people can understand what's going on more, they're less worried about it, and you're ready, you're prepared for what happens. It's like data breaches that we know happen all the time.
Anna, you're part of a public institution, and you have liaisons with the government, with the political system all the time. Are you happy with where the conversations are about responsibility and the? Do you think they're going in the right direction? Should we be having more of them? It seems to me that this is a very scientifically oriented audience, and there are certainly people from the policy world. But maybe this is back to all of you. But let's start with you. But are you happy with where things are going as we move, again, to keep the frontier element of this? Are you happy at the frontier?
Well, I think that more science is needed always to make a better world. That's what I fully believe, of course.
Most people in the audience believe that, yes.
And I think that to involve the people, yes, absolutely, to educate the people. But to educate also multidisciplinarity is absolutely the key. I truly believe it. And to bring human sciences into the field, to understand how the mind of people faced with novelty, to educate them not to fear anything from science and novelty, but to try to better understand. And I think that should be the leading thought. And of course, I think that politicians, they try to do that. They try. But at the same time, yes, we must have secured hospitals, because when we see the disaster it is when a hospital IT attack happens. So I truly believe that we must be optimistic by associating the patients, the people, educate more, and explain, and keep going on explaining. Yeah.
So for a quick fire round, I'll ask each one of you to paint a more optimistic picture of what you are excited about AI enabling in your particular field in the next 5-10 years. When we come back in 5 years, what will you be able to tell us that happened in pathology, genomics, discovery?
For me, I would like to be sure that we have tools to facilitate the work of the pathologist, but at the same time, understanding cancer will come from integrating a lot of data, and also to try to figure out how to prevent cancer. And this is
preventive pathology. Okay.
Yeah.
All right, Janet.
Me, understanding the chemistry and the biology of life, which is we still don't in many different areas, and being able to apply it in medicine for preventative, exactly as you say. And also just being able to use all of this knowledge that we have, frankly, to make the world a better place.
Anna.
Well, it's, I'm coming back to the units of life, right? The cells. And I think Sir Paul mentioned this earlier. I've discussed it last night with John Jumper and also with Demis previously. I think we need the equivalent, we're speaking about AI and what the future might hold. And if we're going to understand life, then we must understand the unit of life. And to understand cells, I think the complexity of biology is so high. that we need the equivalent clean data set like the PDB was for AlphaFold. I think people in the audience would probably agree with me. I was discussing this with Pushmeet earlier as well. We don't have it at the moment. And so what I would like to see over the next 5 years, quite literally, and I'm hoping to be part of that, spearheading that effort, is a moonshot to get a clean perturbation data set
Because
of
you're
human cells.
You're a skeptic of synthetic data,
I am, in the sense that I think
which is not the case with many people here. So
that may be true, and I'm not a computer scientist. I'm a cell biologist, so of course we have I have my biased perspective, so I take that point as well. But I think that, we need a certain amount of real experimentally derived data as the ground truth upon which we could build, potentially by enhancing in the direction of simulations or synthetic data down the road. But I do think that we need sort of a grand, the equivalent of a PDB or a Human Genome Project, a grand challenge, a moonshot, to get enough perturbations.
But wet lab data.
Enough perturbations of human cells in a systematic way, in a clean data set, well controlled, because otherwise we are in a situation where all of us, including myself, have generated millions of cells worth of data, single cell data, other kinds of data, transcriptomic, imaging, that cannot be patched together in a way that would be most meaningful in the way that the PDB was. So we need a clean data set in order to do that. And I, for one, would love in the next 5 years that we actually develop this data set, analyze the data set, and really learn something more about the unit of life, which is our cells.
Well, with that, I will thank everyone and invite everyone to thank
Thank you.
them.